Jose, and hello everyone and thank you for attending today, and also thank you to the committee for the invitation. So today I'm going to talk about the Marie project, a sister project of what I think is the largest citizen science project currently, the Isala project. I'm going to present the Marie project and also a project we are working together with other sisters to um, fill the gap on menstrual health and the relationship with the vaginal microbiome in Switzerland and also beyond. Let's start. So before I go to introducing the Marie project, I'm going to make like a clarification. So what do I mean when I use the term microbiome? I know there is still some debate on, or like some uh, different definitions, but the one I use to is when I talk about the microbiome is I'm referring to the microbiota or the collection or the microbial communities that are living in a particular environment, plus the theater of activity, which involves the environment itself and the interactions between the microbial communities and the host, which also involves physical conditions plus the biochemical or chemical environment. So with this clarification, let's start. So I think for all of you, if you've been hearing the previous presentations, you already know how the vaginal microbiota looks in terms of composition. But for those that maybe just joined, let me just uh, give a brief introduction. So generally, based on the most studied human populations, the vaginal environment is mostly dominated by lactobacilli. And how these bacteria look like? Well, they are rod-shaped, gram-positive bacteria that produce lactic acid, which contributes to the low pH in the vaginal environment, which is a protective feature of this environment. Um, when we talk about the vaginal microbiome, or when you hear the term vaginal microbiome, you might think that this is particularly relevant or only relevant for women or for people that has a vagina. Well, that's in fact not true. This microbiome is key for the health of not only women but also their children and partners. And how so? Well, the previous presentation I think already gave a very detailed explanation of what bacterial vaginosis is. So I'm just going to briefly touch again, just for those that has joined recently or just for this talk. When there is a shift from lactobacilli-dominated vaginal environment to one where there is an overgrowth of a gram-variable group of bacteria, this condition is called bacterial vaginosis and is usually characterized by the presence of Gardnerella, Prevotella, and other anaerobic bacteria. This condition is quite common. The prevalence of this condition ranges from 4 to 70%, 75%, and it depends on the human population that is being studied, even between depends on the country and also inside the country or between the country from different regions. So there are different factors that influence these differences in prevalence rates. And the condition can range from asymptomatic to one that presents vaginal discharge with or without a fishy smell and also presence of itching. Why is this condition important? It's because it increases the risk of STDs, including HIV and HPV, and also is associated with complications during pregnancy, including premature labor and other vaginal conditions. So it's clear that the vaginal microbiome is key, not only for the health of women, but also their children and partners. And so then the question emerged of what factors influence this microbiome. To answer that question, the Isala project was born in 2020. It was launched in March Isala is the name of the first female Belgian physician, and they enroll in 10 days more than 6,000 women that were interested in knowing about the vaginal microbiome or the vaginal microbiota in relationship to their health. And when they received this big amount of participation, I remember discussing with the team behind Isala. They thought that maybe they will not fill the long questionnaire, the health questionnaire that was accompanied before taking or providing the cell sampling swabs, and they were surprised to see that there was a huge participation. So more than 4,000 women filled the questionnaire, a questionnaire that was really long, more than 100 questions. More than 3,000 women provide their cell sampled vaginal swab by post, and the participation age, the age range of participation was quite broad, from 18 years old to 98. So this is really an exciting project, and it was conducted mainly on the Flemish part of Belgium. So among the findings, like among the factors that this study discovered, some might not be surprising. For instance, factors that influence the vaginal microbiome include our age, which is also related to the menstrual cycle, and also to the use of hormonal contraceptives. But for me, something that was very interesting to find was the use of period products as a factor that influences the composition of the vaginal microbiota. And some other factor that was also really interesting to see was having kids or having children. Even if it was a failed pregnancy, this also had an effect, seemed to have a permanent effect on the vaginal microbiome. So maybe, and right now you might be asking, so what about other populations? And this is where the sisterhood comes. So when we think about other populations, we already know, I think, based on also on the previous, on the discussion panel, I think if you heard the panelists discussing, it's clear that there are some populations that are more studied in general in the microbiome field. And here we did a quick look of studies from 2021, July 2021 to 2023, and looking for those studies that were using or studying the vaginal microbiota using amplicon sequencing of the 16S gene, which is a cost-effective technique. And we found that most of the studies come from North America, Europe, and Asia, and only a few studies come from South America and Africa. So there is a knowledge gap that we need to feel to really understand how the vaginal microbiome looks like and which factors influence its composition in different populations if we really want to have therapeutics or solutions for many of the vaginal complaints or vaginal diseases that are associated to the vaginal microbiome. So Isala is when I heard the first time about Isala, I was so excited about this project because of the citizen science component and because it was not only focused on the scientific aspect, it was also focused on the social aspects. It has societal goals like breaking taboos about surrounding vaginal health, encouraging women to talk about different aspects of their health that usually don't talk about. And like me, many other scientists have been inspired in other countries. Initially, it started in Peru with the Laura project, and then in Switzerland with the Marie project. I remember talking with Sandra, which is Sandra Condori, which is the researcher leading and leading the Laura project and coordinating the Isala sisterhood. It was 2021 1 year after Isala was launched, and we were planning to. She already got funding for Laura, and we were planning to get to start Marie in Switzerland. And we were thinking, like, imagine the world full of sister projects. I just pictured what you are seeing right now at that moment in my mind, and I'm really extremely happy to see that this is actually happening. So all of these projects are named after iconic women. In Peru and Switzerland, it was named after the first female physicians in those countries. And these projects, so let's talk now about the Marie project. So these projects are usually, the driving forces of these projects are early career scientists like me, postdoctoral researchers, PhD candidates. For instance, the Leke project, the core or the heart of the Leke project is Josiane, and she's a postdoctoral candidate, Josiane Kempfak. So the path for Marie was a bit difficult. It started when I was transitioning from my PhD. I had finished, and then I immediately moved to motherhood. I remember I was in maternity leave when we started thinking about Marie in Switzerland. So at that time I was almost transitioning to see what were going to be the next steps in my career, and there I met Ethel Mendocilla. She was already finishing her postdoc and moving to project management. So we were actually at that point citizen scientists, and we were trying to see how to launch Marie in Switzerland. So as citizen scientists, we started knocking doors, and it was really difficult to find a place for Marie until we knocked the door. So the Society Gender and Health Unit at the Swiss TPH, led by Sonja Merten. Her group is a multidisciplinary group that has expertise in public health, epidemiology, social scientists, with a focus and with experience in sexual and reproductive health in different contexts. So we found our host lab, and after that, we were lucky to also found other collaborators. We also found Natasha Arora and Meghna Swajambu from the Institute of Forensic Medicine at the University of Zurich. They are the ones that are developing different methods for microbiome detection or for using microbiome biomarkers in crime scenes, including sexual assault. So they are already working with the vaginal microbiome or identification of the vaginal microbiome, and also the penile microbiome. And recently, we also the Marie team has a startup. Jenny Cassetti and Pia Viviani, they are the let's say the core of this startup, and they are really focused on citizen science and science communication. So we have really a great team that covers different aspects of the citizen science nature of the Marie project, and similar to Isala and to the other sister projects. So what are we currently focused on? Currently, we're focused on menstrual health, and we recently got funding from the Swiss Network of International Studies to study what are the impact of menstrual products on the vaginal microbiome. And this proposal, when we planned it and wrote it, it was built up on the findings of Isala. So what they found was that the use of menstrual pad in general as a menstrual product, and also menstrual pad during the last 24 hours before the cell sampling swabs were taken, was associated with beta diversity, alpha diversity, and with the specific members of the vaginal microbiota. So here you can see in green those that are positively or correlated or associated with the use of menstrual pads, either in general or in the last 24 hours before taking the swab. So we have Corynebacterium, Prevotella, we have the Alister and Anaerococcus as positively associated with this, with the use of menstrual pads. And this is also, they are also negatively associated with the presence of Lactobacillus crispatus and Limosilactobacillus. So if you have followed the previous presentations, you know that these two, or particularly Lactobacillus crispatus, is considered, is usually regarded as a beneficial member of the vaginal microbiota. So these were interesting findings, and we united forces. So we have Marie, Laura, Leke, and Isala. Laura and Leke already, well, Isala already launched their citizen science and has already produced results and is launching new studies. Leke also launched her citizen science in Cameroon, and Laura has already done a cross-sectional study in Peru and is planning to launch a longitudinal study this year. So based on these cohorts, we are planning to study the impact of menstrual products on the vaginal microbiota composition with a focus on safety. So our research questions are basically three. One of them, the first one is to understand what are the beliefs, perceptions, and attitudes of participants towards safety of menstrual products, and what are the criteria women consider when choosing the menstrual product they decide to use to manage their menstruation. The second question is to investigate how the use of these menstrual products impact on the vaginal microbiota composition. And the third one is to investigate if knowledge of the vaginal microbiome or the vaginal microbiota, your own microbiota, has an influence on your beliefs, perceptions, attitudes, or decisions towards menstrual products. So how we're going to investigate this? We have three working packages. The first one is really to understand the perceptions of participants through a mixed methods approach that includes health surveys and qualitative approaches. And the second one is to conduct a crossover trial over 6 menstrual cycles, in which participants will try 3 menstrual products: tampon, pads, and cups, in a crossover trial. I'm not going to give specific details of that. It is just for you. You just need to know that participants will wear the 3 products in different order, which is randomly assigned, and then we do comparisons of the vaginal microbiome per product, and also comparisons to the baseline vaginal microbiome before they start the use or the trial of products. And we have a third working package that is called policy influencing. The idea is the objective of this package is to channel the evidence that we gather in the 2 other packages to organizations that are involved or that are involved in menstrual health, not only in policy but also in advocacy. The idea is to raise awareness about the importance of considering the vaginal microbiome when we talk about menstrual health, or when we make decisions that will impact the life of many women in terms of menstruation management. So with this project, we are contributing to achieve 4 out of the 17 Sustainable Development Goals: good health and well-being, gender equality, quality education, and responsible consumption and production. Well, this is the end. Thank you so much for being here today. And I also want to say thank you to the funding agency that makes possible the INVAJA study. I think I didn't mention that INVAJA is an acronym from Improving Menstrual and Vaginal Health for All. I also want to thank all the organizations that are involved in this study. Although a few people contributed to the conceptualization of this study, there is a huge group of researchers, lab technicians, community workers, and the women themselves that make this work possible. Thank you so much.